ในช่วงต่อไปค่ะทุกท่านจะได้รับชมวิดีทัศน์ในหัวข้อ Trends in Quantitative Cellular Imaging for Research and Drug Discovery โดยดร Preston Teng The Imaging Specialist for Perkin-Elmer จากบริษัท Perkin-Elmer จำกัดขอเชิญรับชมค่ะ
Good day, everyone. Thank you for your time and interest to attend today's webinar. I'm Preston, the imaging specialist for Perkin-Elmer Imaging Portfolio in Southeast Asia. And today I'd like to talk about how we can use Perkin-Elmer technology and workflow solution to perform quantitative cellular and in vivo imaging for research and drug discovery. Right. First, I'll talk about how we can use an automated high-content imaging analysis to look at different sample and understand different disease model before moving on to how we can apply in vivo imaging in small animal models, in particular the mouse, to look at the development and characterization as well as assess for the therapeutic efficacy of different disease model. Shown here is actually a quote from the review on high content screening article. High content screening combines the use of automated fluorescent microscopy with quantitative image analysis, and this allows acquisition of unbiased multi-parametric data at a single cell level. For those of you who are not familiar with high content imaging or high content screening, basically it can be defined as we are using an image-based screening method to measure the biological activities in cells. We are not limited to any particular applications when it comes to high content screening, as long as we have the ability to stain and detect the biomarker and protein of interest in our sample of studies. For example, a number of biological questions can be addressed with HCS. We can quantify changes in response of cells to genetic or chemical perturbation, and then identify genes that are required for a specific process. Or we can look at cellular process like differentiation or proliferation, and then using different model of diseases, for example, be it cancer models or infectious disease. And then we can also add different drugs, small molecules and compound to do genetic and chemical profiling to see how the cell responds to this treatment. Shown on this slide is an illustration on how we can get more information from a cellular sample based on high content screening and imaging as compared to conventional method like flow cytometry. So from the flow cytometric plot here, we can see that there are two major populations of cells based on the antibody used to stain them. However, we don't have the phenotypic information as we are not able to visualize how each cell look like in the flow cytometry data. As compared to high content screening, we are able to take image of the samples and then see how the cell looks like. Clearly, you can see that there are 4 different phenotypes of cell within the population, and we have the ability to quantify the parameters within these cells based on high content screening. So this will provide additional information as compared to flow cytometry. And you're able to get more phenotypic data in terms of how the cell looks like and behave under different conditions. Another term that you might have heard or come across is high throughput screening, and this is actually different to high content screening. Typically, a high throughput assay is done using a multiplate reader, whereby we are looking at a single readout per well. However, for high content screening, because we can do multiplexing and stain for different markers and protein, we are able to have multiple readouts per well. Because as shown in the illustration earlier, there could be different phenotypes for a cell in an assay. So we are able to gain more information from the cellular sample. So shown here are some example images of the different sample types that could be imaged using our system. So you're not limited to 2D monoculture like HeLa cells. We also use primary cells or co-culture of stem cells. And this can be extended to more complex 3D models like microtissue, organoids, zebrafish, and even tissue imaging. So this illustrates to you the flexibility that our system offers you, the capability to image different sample types and then analyze them subsequently. Now I would like to introduce our high content imaging systems. We have two instruments, the Opera Phoenix Plus and the Opera CLS. From the picture, you can see that they appear quite similar. And then they are operated by the same operating software called Harmony. In the following slide, I'll explain what are the hardware differences that they have. Shown here on the left is the Opera CLS. It is a single camera system and uses LED as an excitation source. And it is capable of doing confocal imaging and comes with temperature and carbon dioxide control options for live-cell imaging experiments. On the other hand, the Opera Phoenix Plus is a high-end imaging instrument and can be configured with either 1 to 4 cameras and uses laser as an excitation source. We also have this proprietary symphony optics design to suppress crosstalk during fluorescent imaging. And in addition, it can also be fitted with a liquid dispenser to do liquid handling experiments. And in addition to temperature and carbon dioxide control, it also has a humidity control options. So apart from the hardware differences, both systems share quite a number of hardware similarities. For example, they use the same 4.7 megapixel CMOS camera and uses both air and water immersion objective, and they can both do confocal and wide field imaging as well as bright field and digital phase contrast. And they use the same stage for XYZ image acquisition, and a total of 6 different objectives can be fitted on both systems. And they use the same software for image acquisition and data analysis, and both systems can be integrated with automations if there's a need to speed up the handling of the plates during the experiments. So shown here are some example images of the same sample plate taken with different objectives to illustrate what are the details that could be visualized with each magnification. So the objective can range from as low as 1.25 times and all the way up to 63 times magnifications. And the cells are stained with a nuclear dye and the microtubules in green. Next, we would like to demonstrate what is the benefit of using confocal imaging together with the water immersion objective when we are handling complex samples such as a 3D microtissue. So shown here are some examples of images of the same liver microtissue stained with cell mass rate and Hertz, and was acquired with either an air objective or water immersion objective in wide field imaging on the top panel. So clearly you can see that there's a lot of signal scattering across the samples to result in a blurry image, and we could not visualize how this actual sample looks like. So typically we'll apply confocal imaging for thick samples to remove the out of focus light. However, there are some differences between the resolution when we are using either the air objective or the water immersion objective. Clearly you can see here the resolution is greatly improved when confocal imaging is combined with water immersion objective. Moving on, let's focus on the most important part of the workflow, which is high content analysis, and it is done using one software called Harmony, whereby you can perform image acquisition, data visualization, segmentation, and more importantly, quantification in 2D or 3D. Last but not least, we wish to re-emphasize the most important step in the high content imaging workflow, which is image analysis. With our easy-to-use software, you have the ability to convert images to numbers and to knowledge. As a summary, high-content imaging analysis allows us to analyze more samples as compared to conventional microscopy. It not only relies on the interpretation of individual selected images, but also allows statistical analysis of a series of images per treatment condition. And depending on the number of images per well and the number of replicates, it can greatly reduce bias as well as the human error that could possibly arise from analyzing individual microscope images by eye. It also allows to achieve higher physiological relevance as compared to a biochemical assay, for example, that look at the binding interaction of a compound to its target protein. and it is often lacking physiological relevance because the cellular environment is missing. Therefore, high-content imaging is the ideal technology to analyze complex models like 3D microtissues, spheroids, and organoids. And this method and technology will allow us to gain deep insight into the biology. And we can generate hundreds of readouts per cell. So instead of just analyzing parameters like the fluorescent intensity in the flow cytometry, high-content imaging allows us to analyze the phenotype of cells and allows us to generate a phenotypic fingerprint consisting of a detailed morphological description of every cell in the assay. The productivity achieved with Perkin-Elmer high-content imaging systems can be exemplified by more than 1,000 publications between 2010 and 2019. The list of publications is available online and categorized by applications or research areas according to the imaging system used. To wrap up, here's a snapshot of some of our major customers globally, where they have been using Perkin-Elmer's high-content imaging analysis technology to partner them in their discovery of research and drug discovery. With that, I thank you for your attention and welcome any question.
Moving on to in vivo imaging, here's an overview of the presentation which I will introduce about the concept and applications of optical in vivo imaging using mainly mouse models. And optical imaging can be divided into two broad categories: bioluminescence and fluorescence imaging. And Perkin-Elmer offers some examples of how optical imaging can be applied to study and understand different aspects of disease biology. There are certain prerequisites before in vivo imaging can be done, which are like producing optical reporter proteins or dye that we need to introduce them to cell lines, microorganisms, or biomarker of interest so that we can visualize, track, detect, and locate them in these small animal models before performing longitudinal studies. Right. Once the light producing optical reporters agents are established, and the methodology is very simple. We transplanted the labeled cells or inject the fluorescent dye into the animal models and then perform imaging over time before doing data quantification and analysis. Shown here is a schematic that shows how optical in vivo imaging works. Basically, there's a light source produced within an animal. Then you'll get scattered and diffused throughout the tissue, and the intensity of the light source is directly proportional to the intensity of the emission relative to the depth of the source. The CCD camera at the top of the imaging system will then detect and measure the amount of light that passes through in terms of count per pixel. And this way you can quantify the amount of light that can be detected in the animal models. One of the most commonly asked questions is to which mode of imaging should I use? In preclinical setting, I would recommend using bioluminescence whenever possible because it is 1,000 times more sensitive than as compared to fluorescence imaging. Plus, the range of bioluminescent cell lines, reagent, microorganisms, or lentiviral constructs are extensively available to help you to start with your in vivo imaging experiments. Okay, this slide shows the range of optical imaging systems under the IVS series. We have benchtop system for 2D bioluminescence and fluorescence imaging for the output of 3 to 5 mice. And some of these systems have additional X-ray function, so we can have anatomical information on these small animals. In addition, we also have standalone systems such as the IVS Spectrum and Spectrum CT that can do both 2D and 3D imaging. And last but not least, we have a dedicated standalone high-resolution micro-CT imaging system, which is known as the Quantum GX2, which I will not talk about today. Right. No other imaging system has come close to the in vivo sensitivity in terms of bioluminescence imaging. This is because our proprietary IRIS camera can be cooled to minus 90 degrees Celsius, and this allows us to detect to a single cell that is able to produce light within the animal. Okay, and this has been shown in publications. Right? And on here, we have a mouse injected with different cell numbers, and you can see that the amount of light that was detected is directly proportional to the cell number. Since the IB's optical imaging platform was launched in around 1995, it has generated close to 16,000 publications spanning across different research disciplines and disease models. This clearly illustrates the recognition of in vivo imaging and the scientific productivity that can be achieved with Perkin-Elmer imaging technology. And this slide summarizes the essence of in vivo imaging, as we can use it to monitor disease development by performing longitudinal studies through the introduction of label probes, biomarkers, or cell lines. We can also characterize the mechanism, right, to understand the disease biology. And lastly, we can use the animal model and imaging platform to assess for the um, therapeutic efficacy of small molecule drugs, antibodies, and compounds, or even immune cell therapy. So shown here is a slide some time back that shows the compilation of compounds or drug candidates that have been tested or validated preclinically on the IBS imaging platforms in different disease cancer model. From the previous slide, I've shown you a picture of 2D imaging whereby we have a bioluminescent signal represented in a pseudo color that's overlaid onto a snapshot of a mouse similar to what is shown here. Right. In this mouse, we have injected with some labeled cancer cell line in the head. But based on 2D imaging, we cannot tell where these cells are localized in spatial within the animal, as well as in 3D, because anatomical information are lacking or missing. Therefore, if we were to do a 3D tomographic imaging and reconstruction, as well as using X-ray functionality, we can identify additional information such as depth and localization or location of the cells or biomarker and pinpoint them to a particular organ or tissue site to understand the disease biology better. Right. Shown here is a reconstructed images of a mouse in 3D, and then we superimpose a CT data, right, to get additional anatomical information, right. Then clearly you can see that the bioluminescent signal is localized within the brain region of the animal. So if you're interested to find out more about in vivo imaging, please visit our Perkin-Elmer website where we have an extensive online resource library and it contains materials such as white paper, application notes, and more. And we also have this in vivo imaging ebook where you can download online if you're planning to do in vivo imaging to study infectious disease. Right. With that, we've come to the end of today's webinar, and I hope that everyone has learned something from this session. And feel free to contact us if you have any further queries or questions relating to today's presentation. With that, I thank you for your attention and welcome any questions.
ขอขอบคุณดรเพรสตันเตงค่ะ